Tuffaha. I practiced the name and I still got it wrong, or I had to ask. Talking about mutator dynamics that cannot be explained by mutation rates alone.
Okay, hello everyone, thank you for being here. Before I start, I just want to mention that the biggest part of my talk it will be about work in collaboration with David and Brian from the University of Arizona, and the last part is in collaboration with Mridula and Deepa from the Natural Science Center for Biological Sciences in India. So I'm going to start by telling you a hopefully short story. That story comes from my name. So my last name, Tuffaha, in Arabic, means apple. So the first element of my story is an apple tree. The other element of the story is someone that has been around for more than 8 months and studying a PhD with me, and that's my work. This photo actually was taken more than 2 months ago when she sat on her own for the first time. So you can already see that she is exploring her surroundings using her right hand. And she has always preferred her right hand at that time, or her right side in general. So when she's on her back, she would like flip on the side to the right, and like anything, she prefers like picking things that are on her right. So Noor, at that time, was a biased baby. She had a bias because she preferred one side over the other. So let's say now, to the imaginary part, let's say Noor is not only a biased baby, but a biased super baby that can pick apples. If she goes to pick some apples from this tree, what would happen? You can imagine she will start picking from the right side, taking all the ones on the right and just not having access to the ones on the left because she's just more comfortable with the right side. So after a while, she will leave the tree, go back with the apples that she has, leaving the tree with more fruit on the left available, while the right ones are used or taken. And that brings us to today. Today, Noor is more balanced. She can use both of her hands almost equally. And this is one change that happened to her. And the other change is that she is now faster, so she can pick more apples. She can pick apples really faster. So if she now goes to the same tree to pick apples from that, these two changes that happen to her are beneficial, right? So she can now have access to the apples that are on the left that she did not reach before, and she now can pick more apples per unit time. So this is just an analogy of some mutational process, and it is in fact that mutations or organisms can have mutational biases, and these biases can shift in different directions, and there are possible elevations in mutation rates as well. So today I'm going to quickly tell you about the story that we developed in our paper, and it's mainly about the interaction between these two possible changes, the elevations in mutation rates and the shifts in the biases, interaction between those, and how this affects adaptation. And then I'm going to show you some simulation results that validate our theory. And finally, two quick examples that we're working on in cancer. This applies to asexual organisms, and so that applies to cancer and bacterial growth. Okay, so many processes are involved in mutation, but a very important part is DNA repair enzymes. And these are, so when DNA gets replicated, errors happen, and these enzymes, their function is to fix these errors, but they're not 100% accurate, and the errors that they cannot fix are the mutations. And it turns out that these DNA repair enzymes are biased, so they can contribute to not fixing errors from one class of mutations more than the other. And it's also seen that this bias can change because some repair enzymes can lose their function because of a mutation that happened or some environmental that can change, and so they can change or lose their function, which can lead to a change in the bias or an elevation in the mutation rate because the errors will not be fixed as often as before when the enzyme was working well. So a mutator is an organism that now has a higher mutation rate. So whenever I say mutator, I mean an organism with an elevated mutation rate. So one very conventional classification of mutations is the transition-transversion distinction. And I'm sure you already know that every nucleotide in the DNA has one of four possible chemical components. A, G, T, or C. A and G are purines, so they are in one chemical group, and C and T are in another chemical group, and that's pyrimidines. If we go horizontally on this figure, we are in, like the mutation, a mutation that takes us horizontally on this figure means that we did not change the chemical group, and that's called a transition. Going vertically changes the chemical group, and that's a transversion. At any point on this graph, you can see that there is one way of making a transition and two ways of making a transversion. So an unbiased organism would sample twice as many transversions as transitions. Or in other words, the frequency of transitions in an unbiased case should be 1/3, and that's the alpha level that might be referred to. But in nature, organisms are not unbiased, they're in fact biased, and many organisms are actually transition biased. So you see transitions overrepresented in nature, and this level at which the organism is evolving for a while, or historically has been evolving, is what I'm going to refer to as the beta level. But this bias, as I mentioned before, can change if the function of the DNA repair enzyme changes or stops. And the shift in the bias can go up or down. So the first case is to go down to reduce the bias. So you have more transversions being now represented, but transitions are still overrepresented. The other case is to reverse the bias, so now transversions are overrepresented. And these two cases, the reduction and reversal of the bias, can be beneficial because you now have access to the beneficial transversions that were overrepresented before. The third case is a reinforcement of the bias, so you're now sampling even more transitions, and that can be bad for the organism because you have even less access to the beneficial mutations that you need. This new level of the bias after the shift is what I'm going to refer to as the beta prime. And we show in our paper that the interaction between these, the effect of the bias shift can be, the effect of the bias shift is related to the position of beta prime relative to the level of the bias before the shift and the unbiased level alpha. And you can take a look at our theory there if you're interested. But let me go quickly to show you some simulation results of all of that. And so in our simulations, we use an NK fitness landscape. And these landscapes are in fact very high dimensional landscapes, but now I'm just showing a 2D, a 2D example just for convenience. And every point on this landscape represents one possible DNA sequence for the organism, and the height represents the fitness of such organism for each DNA sequence, and the fitness is just a measure of how good this organism, how well this organism does in this environment. So the higher, the fitter, the better. The simulation starts, the population starts in a random place on the landscape, and then mutations start happening. Selection works, does its job, and prefers or favors beneficial mutations that take you uphill and eliminates deleterious mutations that take you downhill. So as time goes, population goes up one of the available hills. And a mutation that took me uphill at some time, in the next generation, reversing it would take me downhill, right? So as time goes, beneficial mutations are used up, and more deleterious mutations will be available for next generations. So as time goes, the beneficial fraction of mutations it goes down, and the deleterious fraction that takes me downhill grows. And you can see this from this figure. As time goes, the mean fitness of the population goes up, so we're going uphill, but the beneficial fraction goes down, so there are less fruits available on the tree as time goes. So in our simulation, we allow the population to evolve at a fixed mutation rate and fixed bias for a while. And when we reach one of these two checkpoints, and that these are when the beneficial fraction reaches 30% or 15%, we initiate an invasion test. And these invasion tests are done by taking 5% of the population, making a change to them, and then see whether this subpopulation invades or not. We repeat that many times, count many times it invades to find the invasion probability. This change that we do to this subpopulation can take one of these three possible forms. So we either keep the bias as it is and increase the mutation rate, or we keep the mutation rate and shift the bias, and then finally, we do both and see the interaction between these effects. I'm going to show you only one figure of the results because we have a lot and I have limited time. So here we allow the population to evolve with 19% transitions, so we are very transition biased. And on the x-axis, you see the mutation rate multiple, so we are multiplying the mutation rate by 1 to 5, etc., up to 1,000. And on the y-axis, you see the invasion probability of this mutator. And this happens at a 15% beneficial fraction. And here you can see the invasion probability of the mutators that have no bias shift, so only the first case. Then we repeat these simulations by doing these mutation rates, but now we shift the bias, and we shift it to a reversal case where transversions are overrepresented now, so only 10% of transitions for the mutator now. And you can see that it has a higher invasion probability as expected by the theory. And if you look at this area here, you can see that at very high mutation rate, because you only have 15% beneficial fraction, so you have many deleterious mutations that you're sampling, and with very high mutation rates, these deleterious mutations that just do not give a chance for the small fraction of beneficial mutations that you have to give you a chance of invasion. But when you shift the bias, you really have a good chance. But again, at very large mutation rates, again, you go to a zero probability. So this is our main result. We also show that in the case of reinforcement, you get it in a lower invasion probability because that's bad for the mechanism, as expected by the theory. So you can check our preprint. It's in bioRxiv, and it's already accepted in the American Naturalist journal. So it's in the proofing process now. So if you're interested, you can see more results there. But in the left time that I have, I just want to highlight some of the applications that we're working on right now. The first one is in human cancer. So we analyzed normal tissue mutations in humans to find the frequency of transitions in these different tissues, and that's the distribution that you hear in different tissues. And you already see that they are transition biased because all the tissues have a higher frequency of transitions than the unbiased level, which is the alpha level 1/3. And then we analyzed cancer human data to also find the frequency of transitions in them. We have two data sets, cosmic and PCOT. And what we hoped to find was a reduction in the bias or a reversal, so going downwards to represent less transitions and more transversions. Because that's the case that corresponds to the fact that cancer outgrows tumor cells. In other words, it's invading the body, right? So we hope to see a reversal or a reduction. And that's kind of what we found. We saw a small reduction. It's a bit different in the two data sets because they have very different forms. But this is actually more reliable because it's very raw data. And while this is a processed kind of data, but anyway, even this has on average, let's say, there are some outliers, but on average, it is a bit reduced, the bias. So that was nice and encouraging. We're now working on finding some more patterns of mutational processes by analyzing more this data in more details. The other application is in bacterial growth. So our collaborators are working on running similar competition experiments to the ones that we simulated. So they also take the 5% frequency for a mutator, and then, but now they have actual mutator that is like having a competition in the lab of E. coli, and they repeat that a few times and like record what happens through time. So we have two mutators, MutY and THNEI. I'm going to only show you these two. We have two more. So in a glucose-rich environment, the MutY almost always wins. So the above figures are the simulation ones, and the below ones are the experimentals. I'm sorry, the resolution is not very good here, but this is the best figure that I have from them right so far. So you have time on the x-axis, mutator frequency on the y-axis, and you see that MutY always wins, both in the simulations and the experiments, while NTHNEI just wins sometimes and fails in the others. So we have a qualitative correspondence between simulations and experiments, but we have a time scale mismatch, so the invasion here is way faster than in the experiments, and we're like trying to figure out the reason for this. But qualitatively, we have the similar result. On the other hand, so this is in a glucose-rich environment. In another environment, we do not have experimental data to compare with, but we have enough. We have the parameters that we need for the simulations, so we run the simulations. And what we see is like in the LB environment, it's like the effect is flipped. So now NTHNEI always wins, and this sometimes wins and sometimes fails. What's the problem here? The problem here is that these two mutators in these two environments have the same mutation rate, the same bias, but they act differently. So clearly, mutation rates and biases are not enough to explain what's going on. So now we're working on understanding what these two environments are doing, what environmental factor is there to explain what's going on and add that to our work. So we have not studied this yet, but we're working on it. So what environment does, when the environment changes, what happens to the landscape is that these peaks either change their height or their position. So what is beneficial in one environment can be a bit deleterious in another. So this changes what how many beneficial mutations are available, and what fraction, how they affect the organism, and so on. And that's, as I said, what we're trying to quantify right now. So in summary, bias shifts, as I said, can be beneficial if they occurred at the right time and in the right direction. And mutators can have an advantage, and this advantage can be elevated if the bias shifts in the right direction. We saw that in cancer, we have a slightly reduced bias, which corresponds to our theory and to the fact that cancer has a selective advantage. And in the end, we saw that environment plays a big role that we're trying to figure out right now, and that contributes for sure in mutator emergence. And of course, in the end, I want to thank the funding agencies, NZERC and the National Institute of General Medical Sciences. And of course, thank my great supervisor, Lindy, and everyone here for listening. Thank you very much.